All right, let's just jump right in. For pretty much all of human history, our DNA, the code of life, was something we could only read. But wow, in just the last decade or so, something that felt like pure science fiction has become a reality. We're talking about the power to actually edit life itself. It's wild.
Just think about this for a second. What if you could find one single typo in a book that's 3 billion letters long and just, fix it? What if you could go in and correct the tiny genetic errors that are responsible for devastating diseases like sickle cell anemia or cystic fibrosis or Huntington's? Well, guess what? This isn't some far-off dream anymore. It's the very real promise of a technology called CRISPR.
And we're not just talking about experiments in a petri dish here. No, what we're witnessing is this absolutely massive shift. We're going from just understanding this tiny, complex biological machine in a lab to actually using it as a therapy in real people. It's just a breathtaking leap from pure theory into a life-changing reality. So you might be asking, how on earth did we get here so fast? I mean, this isn't just a small step forward. This is a complete, total revolution in how we think about and work with genetics.
Okay, so take a look at this. What this chart really shows you is the incredible speed of this revolution. I mean, it's just staggering. Back in 2011, there were only, 3 scientific papers mentioning this key part, Cas9, just 3. Fast forward to 2017, and there are over 2,000. So why the massive explosion? It's simple, really. CRISPR was just so much cheaper, way faster, and more precise than anything we had before it. This thing wasn't a slow burn. It was like a wildfire ripping through research labs all across the globe. All right, so what is this molecular toolkit that has everyone so fired up? Let's pop the hood, so to speak, and see how this incredible little machine actually does its job. So, at its core, CRISPR-Cas9 is actually something scientists borrowed from bacteria. It's their immune system. The best way to think about it is like a search and replace function for DNA. It's got 2 key parts, one part that acts like a guide to find the exact spot you're looking for, and another part that's basically a pair of molecular scissors to make the cut. And here they are, those 2 key components. That big blob you see, that's the Cas9 protein. Those are our molecular scissors. And that little piece, that's the guide RNA. This is the really cool part. It's like the GPS coordinates that we program into the system. It tells the Cas9 scissors exactly where to go, down to the letter. Okay, so here you can see the whole thing in action. That guide RNA, it zips along the DNA until it finds the exact sequence it's programmed to find. And once it locks on, boom, the Cas9 protein makes a clean, precise cut right through the DNA. The key thing to get here is just how precise that cut is. So, now you got a break in the DNA. What happens next? Well, turns out the cell has 2 main ways it can fix that break. The first way is kind of a quick and dirty patch job. The cell just shoves the 2 ends back together. This often creates a tiny error, which is actually super useful if your goal is just to shut a gene off. But the second path? Now this is where the real magic is. If we give the cell a correct piece of DNA to use as a template, it can use that template to perfectly repair the break. It's literally rewriting the genetic code to fix a mistake. Incredible. Okay, so we know how it works, but now scientists have to figure out the best way to actually get this whole toolkit into a cell. And, it turns out there's more than one way to do it. This diagram lays out the 3 main strategies really well. So, option 1, you can send in the DNA blueprint for the CRISPR system. Option 2, you can send in temporary instructions in the form of mRNA. Or, option 3, the most direct approach. You just deliver the whole pre-assembled CRISPR gene, the protein and the guide RNA all ready to go. Now, each of these has its own pros and cons, right? There are trade-offs when it comes to speed, how well it works, and the risk of making accidental edits.
And all of this, it brings us to what is probably the single biggest challenge, the biggest hurdle standing between CRISPR being this amazing tool in a lab and CRISPR being a medicine that helps millions of people. Because here's the reality. We have built the most precise molecular scissors ever known. But honestly, what good are they if you can't get them to the right spot? Getting this CRISPR system into the right cells, in the right part of the body, inside a living, breathing person, that is the huge multi-billion-dollar problem that everyone in this field is racing to solve.
So how are scientists trying to crack this problem? Well, for the most part, their approaches fall into 2 main camps. You can think of them as either special delivery packages or biological Trojan horses. Let's talk about the Trojan horse first. This method actually uses viruses that have been disabled. I mean, think about it. Viruses are absolute pros at getting inside our cells, right? So scientists can hijack that ability to carry the CRISPR system in. It works really, really well, but yeah, it definitely raises some safety questions. Then you have the package approach. This uses tiny, engineered particles like little bubbles of fat to wrap up the CRISPR machinery and deliver it. This way is generally a lot safer, but it's also way less efficient. It's that classic trade-off, power versus safety.
So, as this amazing tech starts to make the journey from the lab and into the clinic, what's the road ahead look like? What are the last big barriers that we still need to get past?
It really boils down to three big things. First, we have to make sure it's precise. We need to be 100% certain that those molecular scissors are only cutting the one spot we tell them to and not making any accidental snips somewhere else. Second, as we've been talking about, is delivery. That's the huge challenge of just getting the tool to the right place. And last, but certainly not least, are the ethical questions. And honestly, those might be the trickiest ones to solve.
And this really brings us to the final and most profound question of all. For the very first time in the history of life on this planet, a species, us, can now consciously steer its own evolution. We literally hold the power to rewrite our own source code. So the question that we all have to face is, what story are we going to choose to write?